Feline Panleukopenia virus, often referred to as FPV, is a highly contagious pathogen that poses a significant threat to both domestic and wild feline populations. The virus targets rapidly dividing cells, with a particular impact on the cells lining the intestinal system. This viral activity leads to inflammation within the intestinal lining, increasing the permeability of the intestinal wall and hindering the absorption of essential nutrients. The study highlights the importance of cobalamin, also known as vitamin B12, which is crucial for several key enzyme systems involved in the synthesis of nucleic acids and the production of blood cells. Cobalamin is absorbed from food through a complex process involving receptors in the intestines. In cats, the pancreas is the exclusive site for the production of intrinsic factor, a protein necessary for cobalamin absorption in the final section of the small intestine, called the ileum. Previous research has indicated that reduced levels of cobalamin can be associated with changes in the bacterial populations within the small intestine or with gastrointestinal diseases, suggesting it could be a valuable marker for assessing the extent of intestinal damage caused by FPV. Copper and zinc are trace elements absorbed by the intestinal epithelial cells in cats and serve as essential components or cofactors for numerous proteins vital for various bodily functions. Research in humans has linked these trace elements and their ratio to conditions like gastrointestinal inflammation, impaired immune function, increased oxidative stress, and malnutrition. Recent studies in dogs have also suggested that trace elements can be reliable indicators of the likely progression of inflammation. Therefore, investigating the prognostic value of serum concentrations of these trace elements in cats with FPV appear to be a promising area for enhancing routine veterinary practice. This study highlights that cats that do not survive feline panleukopenia have significantly higher concentrations of copper and serum amyloid A and significantly lower concentrations of zinc and cobalamin compared to cats that do survive. Furthermore, serum amyloid A and cobalamin demonstrated remarkable prognostic capabilities. Copper also showed good prognostic potential, and the assessment of zinc concentrations was promising for predicting survival. These biomarkers offer a potentially cost-effective way to predict the likely outcome for cats with FPV at the time of hospital admission.